Hello everyone. In previous video, we have studied that immunity is ability of our body to defend against the pathogens. Part of this ability we possess since birth and it is known as innate immunity. If you haven't watched that video, the link for the same is provided in the description box below. After birth, as we get exposed to the different types of pathogens, our immune cells, that is lymphocytes, get activated against the specific invading organisms and protect us from them. Since lymphocytes acquire this ability on exposure to the specific pathogens, this type of immunity that develops after birth is called as acquired immunity. And today we shall discuss some general aspects of acquired immunity. But before we start, for those who are new to this channel, this is Pratima from Planet Physiology. Well, let us begin with the features of acquired immunity, which is also known as adaptive immunity. As mentioned in the beginning, acquired immunity develops after birth and it is very specific in nature. That means it develops against the specific pathogens that invade the body. For example, immunity against measles, mumps, chickenpox. So, acquired immunity develops only on exposure to the specific pathogens. It is conferred by lymphocytes which respond either by forming antibodies or activated T cells. Protection offered by acquired immunity is long-lasting and sometimes may be lifelong, as in case of measles. The last point is that acquired immunity is phylogenetically newer in development. Before we delve into the topic of acquired immunity, you must have a clear understanding of the terms antigen and antibody. Antigens are the specific chemical compounds that evoke immune response. They are usually protein or polysaccharide in nature with molecular weight more than 8,000. They contain certain recurring molecular groups called as epitopes. Epitopes impart antigenicity, or we can say that they are antigenic in nature. But many a times, some low molecular weight substances also act as antigens and they are called as haptens. Usually, haptens combine with some antigenic substance to evoke immune response. Examples of hapten include pollen grains, dust particles, animal dandruff, etc. The second terminology often associated with immunity is antibody. Antibodies are also called as immunoglobulins because they belong to globulin family of plasma proteins and are associated with immunity. The word immunoglobulin is often abbreviated as Ig. So, antibodies or immunoglobulins are the proteins synthesized by the lymphocytes in response to antigens, and these antibodies are specific for specific antigens. Same is represented in this picture. Antibodies against measles can attack only measles virus but not the chickenpox or the mumps. Also, if a particular pathogen invades the body for the first time, our body will not have antibodies against that pathogen but will develop them in due course of time. Now let us study how lymphocytes develop and acquire ability to detect specific pathogens. Pluripotent hemopoietic stem cell is the precursor for all types of blood cells. They give rise to two cell lineage, lymphoid and myeloid series. Lymphoid stem cells differentiate to form B and T lymphocytes, but these lymphocytes are not capable of forming antibodies or activated T lymphocytes until they are further differentiated in the processing structures. Let us study this process of differentiation. From bone marrow, those lymphocytes which are destined to form activated T cells migrate to thymus for pre-processing. Hence, they are called as T lymphocytes, where T stands for thymus, the organ of pre-processing. The lymphocytes which are destined to form antibodies are pre-processed in liver during mid-fetal life and later in bone marrow. Actually, these cells were first discovered to be processed in special structure called as bursa of Fabricius in birds, and hence the cells were called as B lymphocytes to represent bursa. But humans lack bursa, and liver and bone marrow act as bursal equivalents. These processed lymphocytes then enter the circulation and settle in various lymphoid structures like spleen, thymus, tonsils and other lymph nodes throughout the body. So the lymphoid organs possess both B as well as T lymphocytes. Now you may get a question, what is meant by pre-processing and what is its need? Let us understand with the example of defense system of our country. The soldiers who are destined to protect land are trained in army institutes like NDA, while those who are destined to protect sea get their training in various naval academies. In the same way, T lymphocytes are trained in thymus and B lymphocytes in liver and bone marrow. In these processing organs, especially in thymus, lymphocytes divide rapidly and also develop diversity to react against different specific antigens. Here they are also exposed to almost all the self-antigens, that is various proteins on the different body cells. Those lymphocytes which react to these self-antigens are phagocytosed and removed, and only those lymphocytes which do not react with our own antigens are allowed to remain. Thus, lymphocytes are basically trained to differentiate our own antigens from the foreign antigens so that they will not evoke any immune response to our own proteins, but at the same time are capable of reacting against foreign antigens. Similar to the defense posting after successful completion of training, these lymphocytes are posted at various checkposts in the body, that is lymphoid organs, to protect the body against invading organisms. Most of the pre-processing of T lymphocytes occur in late fetal life and continues till few months after birth. Hence, the size of thymus at this age is quite large, but gradually regresses later in the life, which is just sufficient to maintain T lymphocytes. Now let us study how lymphocytes react to the pathogens. Whenever lymphocytes come across any foreign antigen, it binds with those antigens. This binding indicates that lymphocyte has recognized the antigen as foreign antigen. This encounter and recognition of foreign antigen forms first step in the immune response. Usually both B as well as T lymphocytes are simultaneously activated by the same antigen, and macrophages play important role in it. We shall study the details of their activation process in a separate session for B and T cell immunity. Now the next step is lymphocyte activation. It is nothing but multiplication of that specific lymphocyte which has detected the antigen to form clones of it. So the population of lymphocytes produced from the single activated lymphocyte forms a clone, and all the lymphocytes in a single clone are alike, which are capable of forming a specific type of antibodies or activated T cells against the invading organism. Then the third step is attacking the invading organism. The products of these activated lymphocytes attack and destroy the invading antigens as well as antigen-infected cells. So, if you have understood this mechanism of specific immune response, it must be clear that lymphocytes get activated only when they are exposed to the foreign antigens for the first time. Hence, they need a longer time to identify and attack the antigen. This response of immune cells during their first exposure to the antigen is called as primary response. But after exposure to the specific antigen, small number of activated lymphocytes persist as memory cells. These memory cells disperse throughout the body and get lodged in the various parts of the body, especially near the routes of the antigen entry like respiratory and gastrointestinal tract. During subsequent exposure of the body to the same antigen, memory cells are immediately converted to the effector cells, which immediately attack and remove the antigen from the body. So this rapid, strong and long-lasting response by the memory cells is known as secondary response. This graph shows comparison of primary and secondary response in terms of rapidity of the onset, intensity and duration of the immune response. This ability of immune cells to produce accelerated response is another key feature of acquired immunity, and this knowledge forms the basis for vaccination or immunization. Details of vaccination we shall study in a separate session. So let us quickly summarize the important points of acquired immunity. As name suggests, it develops after birth in response to exposure of the body to the pathogens. It is conferred by lymphocytes. Lymphocytes develop capability to identify millions of different types of antigens during pre-processing. B lymphocytes are pre-processed in fetal liver and bone marrow, whereas T lymphocytes are pre-processed in thymus. Particular antigen activates only specific lymphocytes, and hence the response is highly specific in nature. This immune response is a bit slower, weaker and short-lasting for the first exposure to the antigens, and it is known as primary response. During primary response, memory cells are formed which help in quick, stronger and long-lasting response on subsequent exposure of the same antigen. This is known as secondary response. It also forms basis for vaccination. So that's all for this video. Thank you for watching and see you in the next video. If you enjoy my presentations, press the like button and share it with your friends. For more such videos, subscribe my channel and click the bell icon. Thank you for watching and see you in the next video.